Hello everyone, welcome back to my channel. In the last section, we have discussed about the thyroid gland, its gross anatomy in detail. So today we'll discuss about the histology of thyroid gland and about the thyroid hormone synthesis. Thyroid gland has got two capsules. We have already discussed that is inner true capsule and the outer false capsule. The true capsule sends in septa towards the interior of the gland, dividing the gland into many lobules. So the septa divides into many lobules. Each lobule contains aggregates of follicles. So each lobule is a collection of follicles, that is an aggregation of follicles, and each follicle is made up of many follicular cells that rest on basement membrane. So the capsule sends septa inside the gland, which divides into many lobules, and each lobule has got aggregation of follicles, and each follicle is made up of follicular cells which rest on basement membrane. You can also get parafollicular cells or C cells. These are present in between the follicles, or these are present in between the follicular cell and the basement membrane. And the stroma surrounding the follicle is made up of connective tissue, lymphatic, and also has got blood vessels. So this is the histology of thyroid gland. Each follicle has got a cavity which is filled by a homogeneous material that is known as colloid. So that's all about the follicular cells and C cells. So we'll see in detail about the follicular cell. The follicular cell has got, in the electron microscopic view, it has got a granulous endoplasmic reticulum, then Golgi complex, secretory vacuoles, lysosome, and also it has got apical microvilli. The follicular cells vary in shape depending upon the activity of the thyroid gland. In a moderately active gland, the follicular cell is cuboidal in shape. It is cuboidal in shape with moderate amount of colloid. Whereas in an inactive gland, the follicular cell is squamous in shape with larger amount of colloid. Whereas in a highly active gland, it is columnar in shape. It is columnar in shape with scanty amount of colloid. So depending upon the activity of the thyroid gland, the follicular cell's shape varies. That is, in a moderately active gland, it is cuboidal in shape with a moderate amount of colloid. In an inactive gland, it is squamous in shape with a large amount of colloid, and in a highly active gland, it is columnar in shape with little amount of colloid. Now, the activity of the follicular cells is influenced by thyroid stimulating hormone, that is TSH, which is secreted by the pituitary gland. And the follicular cells produce mainly two hormones, that is T3 and T4. T3 is triiodothyronine and T4 is tetraiodothyronine, and T4 is also known as thyroxine. So these are the main hormones of the thyroid gland. Now we will see how the hormones are synthesized. So synthesis of thyroid hormones take place in two phases. In the first phase, thyroglobulin, which is a glycoprotein, is produced by endoplasmic reticulum, and it is packed into secretory vacuoles of the Golgi complex and then released along the apical microvilli into the follicular cavity. And here it combines with the iodine, and then colloid is formed. That's how colloid is formed. So colloid is iodinated thyroglobulin. That is the first phase, and this process is known as exocytosis. Exocytosis. Whereas in the second phase, this iodinated thyroglobulin re-enters the thyroid follicular cell. This is known as endocytosis. Endocytosis. And then this iodinated thyroglobulin is acted upon by enzymes that is released by the lysosome, and then T3 and T4 are produced, and these are released into the blood. So that's how thyroid hormone synthesis takes place. That is, it takes place through two phases, that is exocytosis and endocytosis. While during the exocytosis, thyroglobulin enters into the follicular cavity, and where it combines with iodine to form the colloid, that is the iodine and thyroglobulin. Whereas in the endocytosis, it re-enters into the follicular cell, and with the action of the enzymes released by the lysosome, T3 and T4 are produced and then they are secreted into the blood. That's how thyroid hormone is synthesized. This is the activity of the follicular cell. Whereas the C cells or the parafollicular cells, that also contains Golgi complex, mitochondria, secretory vacuoles, lysosomes. So these C cells produce the hormone known as thyrocalcitonin. Thyrocalcitonin. So this comes into play. The calcium level in the blood is high. So when the calcium level in the blood is high, this is produced, and it lowers the calcium level by suppressing the release of calcium ions from the bones. And it has also got, this T cells has also got a structure similar to the follicular cells. That is, it has got mitochondria, Golgi complex, lysosomes, secretory vacuoles. And we have already discussed about the hyperthyroidism and hypothyroidism in the previous section. So hyperthyroidism is a condition in which the T3 and T4 are high plus TSH is low, whereas in hypothyroidism T3 and T4 are low and TSH is high. This is a hematocele and eosin stained section of thyroid gland. You can see the thyroid follicular cells and then parafollicular cells and then the stroma. Histology of thyroid gland is a favorite question for the exams. So I hope you have understood this short topic, that is the histology of thyroid gland and the thyroid hormone synthesis.